Hello students, today we'll be taking up TCA cycle, tricarboxylic acid cycle. So it is also known as KREBS cycle, TCA cycle, citric acid cycle or tricarboxylic acid cycle. So we will be taking up introduction, then we will talk about the site where it is generated, some of the important steps, enzymes involved in TCA cycle, energetics and significance. Tricarboxylic acid cycle. So the introduction would be like this. TCA cycle or tricarboxylic acid cycle or KREBS cycle takes place under aerobic conditions. Aerobic conditions mean in presence of oxygen. So under aerobic conditions, cells break down glucose into pyruvate. Pyruvate is obtained after glycolysis of glucose under aerobic conditions. So what happens to pyruvate? Pyruvate gets oxidized to carbon dioxide and water under aerobic conditions. This is known as respiration by biochemists. So there are three stages of this cellular respiration. Number one would be conversion of pyruvate to acetyl coenzyme A and carbon dioxide. The second stage would be tricarboxylic acid cycle. It also means degradation or breakdown or catabolism of acetyl coenzyme A. The third one would be electron transport chain and oxidative phosphorylations. These are the three stages of cellular respiration. So in the first stage of what happens is pyruvate, which is obtained from glycolysis, that is degradation of glucose. Pyruvate binds with coenzyme A and NAD is the coenzyme. The enzyme pyruvate dehydrogenase complex converts pyruvate to acetyl coenzyme A, carbon dioxide and NADH. So this is in fact oxidative decarboxylation of pyruvate. This reaction is being carried out by pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex involves a multi-enzyme system, large multi-enzyme system comprises of three enzymes: pyruvate decarboxylase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase, and it also includes five coenzymes: TPP, lipoic acid, FAD, NAD, and coenzyme A. Now here you can see from these graphics, pyruvate is obtained from glycolysis in the cytoplasm by degradation of carbohydrates or glucose, you can say, by glycolysis under aerobic conditions. The pyruvate can also be obtained from degradation of fatty acids. Pyruvate can also be obtained from degradation of amino acids. So what happens is pyruvate formed in the cytoplasm enters into the mitochondria using a symphoter. Now pyruvate undergoes oxidative decarboxylation to acetyl coenzyme A under the influence of pyruvate dehydrogenase complex, which we have seen in the previous video. Now what happens is acetyl coenzyme A has to enter into TCA cycle. It gets catabolized or degraded to carbon dioxide, water, and with release of energy. Now TCA cycle, you remember, it takes place within the mitochondria. Now excess of acetyl coenzyme A is also obtained from fatty acid synthesis. It is also used for ketone body synthesis. It is also used for cholesterol synthesis, and it also enters into TCA cycle. Now what is TCA cycle? TCA cycle is a cyclic metabolic reaction, one of the pathways of carbohydrate metabolism. This is TCA cycle. So there are around 8 reactions in this particular TCA cycle. Reactions take place in a cyclic manner. The reaction begins with oxaloacetate, giving rise to citrate. Next is isostrate, followed by alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, and malate, and again oxaloacetate is regenerated, so it is a cyclic reaction. So it involves 8 reactions catalyzed by different enzymes. First reaction catalyzed by citrate synthase, second one aconitase, third one isocitrate dehydrogenase, next alpha keto dehydrogenase, next succinyl coenzyme synthetase, next succinate dehydrogenase, next would be fumarase, and lastly malate dehydrogenase. So it is at these very steps we get ATP, we get GTP, we get NADH, we get carbon dioxide and water. So talking about the enzymes of TCA cycle, the enzymes of TCA cycle are located within the mitochondria. Here you can see the structure of mitochondria. It is a boat-shaped structure known as powerhouse of the cell. It has an inner boundary membrane. This is an intermembrane compartment. This is the matrix. This is outer boundary membrane, and these are crusty. These very structures are crusty. Here you find this. So mitochondrial this TCA enzymes are very much located in the mitochondria. Now in the inner mitochondrial membrane, we find two important enzymes, aconitase and succinate dehydrogenase. Most of the enzymes involved in TCA are located within the matrix part here. That is citrate synthase, isocitrate dehydrogenase, alpha dehydrogenase complex, succinyl coenzyme synthetase, fumarase, malate dehydrogenase, and PD complex. Now you can see the cycle. The first reaction starts with oxaloacetate. This is oxaloacetate under the influence of enzyme citrate synthetase, also known as citrate synthase. This is acetyl coenzyme A. It condenses with oxaloacetate to give you the first product in the reaction, that is citrate or citric acid. Here you find 3 carboxylic acid groups. That is why it is also known as tricarboxylic acid cycle. Next reaction is dehydration reaction catalyzed by aconitase. You get cis aconitate. This is a kind of isomerization reaction. Next reaction is hydration. Water molecule is removed over here. Under the influence of enzyme aconitase, you get isocitrate. Then here oxidation takes place. NAD plus is the coenzyme. It gets reduced to NADH, and reaction is being catalyzed by isocitrate dehydrogenase. You get molecule of carbon dioxide here. This is oxidative decarboxylation, removal of carbon dioxide. Next you have alpha ketoglutarate is the product. Enzyme is alpha ketoglutarate dehydrogenase complex. It is similar to pyruvate dehydrogenase complex. Here also oxidation takes place. Na from NAD plus you get NADH is the reduced form. You lose one molecule of carbon dioxide. Then you get succinyl coenzyme A. Here GTP is involved. GDP generates GTP. GTP means guanosine triphosphate. It is a high energy phosphate just like ATP. So since succinate is generated, we have an enzyme succinyl coenzyme synthetase enzyme. So succinate gets converted to fumarate. FAD is used as cofactor. Here you get FADH2 succinate dehydrogenase. Finally, it is being catalyzed by fumarase enzyme. You lose a molecule of water. We get malate. Malate is subjected to oxidation. NAD gets converted to NADH. Under the influence of malate dehydrogenase, you get oxaloacetate is regenerated again. So it is a cyclic reaction. Actually, it is catabolism or degradation of acetyl coenzyme A. Acetyl coenzyme is a 2-carbon unit. It gets converted to carbon dioxide and water. Reaction looks to be very simple, but practically it has to take this cyclic shape. This has to carry out this 8-step reaction process to get acetyl coenzyme A completely converted to carbon dioxide, water, and with release of energy. Some of the vitamins are involved in TCA cycle which generate energy. Thiamine acts as a coenzyme, riboflavin, niacin, pantothenic acid. Thiamine acts as a coenzyme for alpha ketoglutarate dehydrogenase. Riboflavin acts as a coenzyme FAD for succinate dehydrogenase. Similarly, niacin is an electron acceptor for isocitrate dehydrogenase, alpha keto dehydrogenase complex, and malate dehydrogenase. Pantothenic acid is a coenzyme attached to active carboxylic acid residues, that is acetyl coenzyme A and succinyl coenzyme. This is the role of these vitamins. Now regulation. Citrate synthase enzyme, the first ever enzyme which catalyzes the conversion of oxaloacetate to citrate, is being inhibited by ATP, NADH and acetyl coenzyme A and succinyl coenzyme A. Whenever there is an increase in the level of NADH or acetyl coenzyme A or succinyl coenzyme A, that leads to inhibition of citrate synthase enzymes. Here it is being regulated. Next is isocitrate dehydrogenase can be activated if there is a high ratio of ADP. It can be activated by ADP. It is inhibited by ATP and NADH. So that depends upon the situation. So alpha ketoglutarate dehydrogenase complex gets inhibited when there is enhanced level of succinyl coenzyme and NADH. Similarly, ADP plays a very important role in TCA cycle. If we have sufficient levels of ADP, because they are needed for oxidation coupled phosphorylation of ADP to ATP, NADH and FADH2 through electron transport chain. Otherwise, it is stopped. You need to have a very good level of ADP in the reaction. Otherwise, this electron transport chain would get stopped. So the accumulation of NADH and FADH also very important, because this would lead to inhibition of all these enzymes and limit the supply of NAD plus and FAD. So these very steps are important for the regulation of TCA cycle. So next we can talk about energetics. How many ATPs are generated? 12 ATPs are generated per molecule of glucose. So in this reaction, isocitrate is converted to alpha ketoglutarate. NADH generated, that generates 3 ATPs under oxidative electron transport chain. From alpha ketoglutarate to succinyl coenzyme A, one NADH molecule is generated, it gives 3 ATPs. Succinyl coenzyme A to succinate, you get GTP, means 1 ATP. Succinate to fumarate, you get FADH to one molecule, it generates 2 ATPs. From malate to oxaloacetate, it generates NADH, it gives again 3 ATPs. So these are the steps where NADH is generated, NADH is generated, and NADH is generated. One NADH upon oxidation electron transport chain generates 3 ATPs. So total you have now 12 ATPs. Thank you.